So once we have the cells we want, including genetic modification if that was required in development of the cell therapy, we now need to grow more of those cells to reach our therapeutic dose. Now, you already learned in Unit 2 that cells need a number of things to grow and proliferate in vivo inside the body, and then we need to be able to provide those outside the body for manufacturing. Let's learn more about how we actually provide those components. As a reminder, cells are made up of proteins, DNA, RNA, lipids, sugars, and other components. And all of these are built from the main basic building blocks of life: hydrogen, oxygen, nitrogen, carbon, phosphorus, sulfur, and then also inorganic salts and trace metals. But cells also require energy to grow and divide. We learned that the cellular metabolism has been optimized in life to occur at specific temperatures and pH on Earth. We need to be able to provide all of these adequately to care for our cells. And most of these cells' needs are supplied by the cell culture medium. This is a liquid or broth that is designed to grow the cells, to promote their proliferation, at least at certain steps, and contains key sources of these elemental needs of the cells. As an example, glucose is a common source of both the carbon and the energy. It's processed through glycolysis and the TCA cycle to produce both energy and the building blocks for several cellular components. Therefore, glucose, or a suitable replacement of glucose, will be a key component of any cell culture medium. And you might find for your cell type that there might be a low glucose medium or a high glucose medium. The medium will also be able to provide a source of the essential amino acids that eukaryotic cells can't make themselves, right? We eat to live, and we actually have to provide those nutrients into the cell culture media for those cells to proliferate and culture. So when the specific needs of a cell are not well understood, we don't know exactly what nutrients make that cell proliferate or in what concentration, we often add complex mixtures of animal or plant proteins. Fetal bovine serum or calf serum is often used, certainly at the developmental stage, and proteins that come from plants like soy hydrolysate are also used. But remember that these are choices because we don't know exactly what the cells need. A key challenge here is that these materials are not well defined. Right? They can vary from batch to batch. Fetal bovine serum can vary from lot to lot, and this can actually affect a lot of the challenges in reproducing your results, as well as create a risk of contamination. As we better understand the needs of cells, we can move towards more defined materials to apply these essential amino acids. So when you hear about people working with defined media or animal component-free media, they're working to provide these nutrients without the complexity of using an animal or plant-based source. Finally, depending on the cell type, additional growth factors or stimulatory factors, such as interleukin-2 shown here, may be required to activate or differentiate the cells as a way of developing the cells that are released to the patient as a product. These components would be added to the cell culture medium as needed, so you can think about these as part of the cocktail of that complete medium solution. All right, so we're most of the way towards giving the cells what they need to grow. Now let's talk about the oxygen that cells require. So oxygen can dissolve into liquids such as water, and the equilibrium concentration of oxygen that can dissolve in a liquid depends on a few things. The type of liquid, whether it's water, whether it's a different type of solvent, the partial pressure of oxygen in the gas that's above that liquid. So here, the gas that's in the air has a certain concentration of oxygen within it, and we can calculate from that the partial pressure of oxygen. And the temperature. So at a high level, what this means is that we can supply oxygen through the atmosphere, from the atmosphere, and have that come into contact with the cell culture medium to provide oxygen to the cells themselves. Now, the rate at which oxygen will dissolve into that medium is also known as the oxygen transfer rate. And that's going to depend on the physical parameters of the culture system and the partial pressure of oxygen in the air around that cell culture medium and the current concentration of oxygen that's already in the cell culture medium. Of course, cells will also be consuming the oxygen at a specific rate, and we call that the oxygen uptake rate, or OUR. That's related to how much oxygen each cell consumes and also the number of cells in the culture. So your cell count here is very relevant and important. Now that we understand how to provide the basic building blocks and the energy for cell growth, we have to also think about how we can control the other parameters that affect the culture, such as temperature and pH. Now, in the case of temperature, it's no surprise that most human and mammalian cells are best supported by temperatures in the range of 36 to 37 degrees. I mean, after all, that's body temperature. So we need a way to both measure and control the temperature of the system for optimal cell growth. And ideally, we'd like to keep that constant. This is an important consideration as, for example, you're taking cells in and out of the incubator. And in the case of pH, most human and mammalian cells are supported by pH in the range of 7 to 7.4, so neutral or slightly more basic than neutral. In the case of pH, think about the fact that one of the byproducts of the cells being viable and having an active metabolism is that the cells will produce lactic acid. And over time, and especially at high cell count, this is actually going to acidify the culture medium. And so we need to either remove the lactic acid from the solution to maintain the pH or find a way to compensate for that natural production of acid by the cells as they're growing and they're increasing in number. So removal of lactic acid is often not possible or practical. And so the way that we tend to compensate for the production of acid in the culture is to provide a specific salt that buffers or maintains the pH of the solution. One of the most common buffers that's used is bicarbonate. And this utilizes equilibrium between the atmospheric CO2, the carbon dioxide that we can provide in the gases that are surrounding the culture chamber, and the dissolved carbonate to maintain a target pH value. So this is an easy way to buffer the solution, is adding the bicarbonate. But ideally, we would measure the pH of the culture medium over time and adjust that atmospheric carbon dioxide accordingly. This isn't always possible, but this would be the aspiration of a bioreactor design. We also need to make sure that the cell culture system is appropriate for our target cells of interest. For example, some types of cells, like keratinocytes, only grow and proliferate when they're attached to a surface. They're anchorage dependent, and so we need to have cell culture equipment that will support that need for attachment in order for the cells to proliferate, such as a feeder flask that has high surface area on the bottom of the flask, and that's where the cells will attach and grow in number. But conversely, other cell types, like T cells, can grow and proliferate when suspended in solution, and therefore the instruments in the culture vessels that are used to grow those cell types will look different and not depend on strong interactions between the cells and the culture materials, like the vessel surfaces. And now finally, we need to make sure that the systems we use to grow and to transfer the cells maintain sterility, and they don't provide opportunities to introduce unwanted microorganisms like bacteria or yeast or viruses that could come from what we're adding to the cells or even from ourselves, because these conditions that proliferate the cells that we're trying to make for cell therapy are also ripe for proliferation of these other organisms. Now, the concept of keeping out unwanted contaminants is known as aseptic processing. You may also hear this referred to as sterile technique or sterile cell culture. But at the heart of it, what this means is that the cell culture systems have to be closed to the environment that might add contaminants into the system to keep those contaminants from getting in. So this is really a defensive approach so that we don't have to add anything to actually remove those unwanted organisms. So you may use and tend to use a closed container, a capped flask, and there may be a filter on that flask cap in order to allow exchange of gases. But if we do have to open the system, for example, you open the flask to feed the cells, you're exposing to the environment, and therefore you'll have to do this in a highly controlled environment like the one in a biosafety cabinet. The key point here is all the aspects that we've discussed need to be optimized and then also controlled to allow us to expand or proliferate the cells in culture to reach a target dose for therapeutic administration. Exactly how we control the culture conditions to encourage cell growth depends on the scale of the cell culture, what is the total cell volume that we need, and what are the technologies available to support that, and also the types of cells that we're growing. Are they adherent? Are they suspended? What kind of conditions do they require? So in the next few sections, we'll take a look at examples of the equipment that is used to control the environment and to encourage the cell proliferation and growth.